Okay, welcome to Biology 1100. This will be a lecture about how to prepare for um, writing a report and doing an experiment in class. So it's basically the term project lecture today. And I'd like to talk about experimental design, the equipment that will be available, and statistics, and even just starting from the very beginning. How do we start doing research in the first place? Well, first of all, you have to be interested in something, and in this case, we're interested in animal behavior, and maybe more specifically animal behavioral responses to the environment. And it's of extreme importance in today's environment because it's changing for a lot of animals. So, for example, we're changing the acidity of the ocean, and that can affect animals that use carbon in calcium carbonate shells. So the tolerances of animals to different abiotic variables are important. So the importance of this may be the tolerance of animals to abiotic conditions, which may be changing. You know, things like salinity, acidity, temperature, um, intertidal composition. So these things are changing. So it's always good to do experiments in areas that you're interested in. You know, areas of interest. That's quite important. It must be something that triggers your interest. You know, somebody told me a long time ago, you've got to do something that gives you a fire in your belly. That's how he said it. And, you know, when I went on a field course in Costa Rica into the jungle, that created this huge fire. And from then on, I was so interested in ecology. And so, yeah, I'll try to start at the beginning, which is making observations. So you've got your area of interest, and then you make observations. And you've done that by doing your literature report. You can also go out in the field and make your own observations. You can talk to other researchers in the same field. And it's important to know what is already known. So the reason to make observation is to find out what is already known about the animal. And what questions need to be asked. With respect to changing environments, perhaps you're concerned about conservation of the animals, or perhaps you're just interested from a purely scientific interest uh, direction, and that's perfectly okay. Pure research for no application reasons is perfectly okay. Just increasing knowledge is a noble thing. So, what questions need to be asked? Well, maybe you're going to ask a question about the behavioral response of animals, directing you towards what we're doing in the class, the behavioral response of animals to certain variables. So as an example, I might ask the question, how does temperature affect brine shrimp metabolism? That might be my question. Now, can I measure metabolism directly? I probably can't unless I have really skookum equipment. Um, but I can measure their movement. So increased metabolism would be related to their movement. So that is something I can measure. So their response must be something you can measure. That would be considered, what is it? A measured response kind of experiment. Wait, there's a name for it, I think, here in your lab manual. Oh yeah, measured response and preference response. So this would be a measured response. The other style of questioning is called a preference response. I just call it a choice experiment. And so that's the question is a little bit different. The question is, what kind of environment do these organisms prefer? What would their choice be? So the question is, what levels of temperature do the animals prefer? So question 1, that leads to one type of experiment. Question 2, that leads to a different kind of experiment. So let's look at our hypotheses. We've asked the question. The next step is always to form hypotheses. It's very important. So for question 1, we give the animals, or no, sorry, that's not how I should say that. We place the animals in 3, for this purpose of our experiment, we'll choose 3, in 3 different environments. And we have to be able to measure their response in these environments. So say we have 3 different temperatures. We have to choose a temperature that won't kill the animal. That's very important. Neither freeze it nor boil it. So let's say we go 5 degrees Celsius, 15 degrees Celsius, and 25 degrees Celsius. 3 different environments. One important aspect of experimentation is that we don't just use one sample one time. Instead, we use replicates. So I would recommend that you use at least 3 replicates. These are Petri dishes, by the way. 3 replicates. They each have the different temperatures, so these will all be 5 degrees Celsius. And you can do more, it just depends on the time that you have. 3 is usually plenty. And we'll measure their response in some way. So If we're measuring movement, um, maybe we get 3 different distances of movement. For our first replicate, maybe it travels 15 centimeters. Maybe the second one travels 25 centimeters. And maybe the third one 20 centimeters. So we need to get the average. And the average in this case will be 20 centimeters. And we'll get the average for all 3 treatments. So that'll be treatment 1, treatment 2. Let's just pretend it's 60 centimeters on average. And treatment 3 will be, who knows, maybe 10. We always use averages. So our hypothesis will be, didn't want to do that. Our hypothesis will be, hang on, I would like to insert another lined paper. So our hypothesis will be a null hypothesis and an alternative hypothesis. So in science, we're disputing the null hypothesis, if we can. That's what we're trying to show. And the null hypothesis means that there is nothing interesting going on. Nothing interesting going on, which would mean that mean number 1 is equal to mean number 2, which is equal to mean number 3, statistically. But our alternative hypothesis will be that they are not equal. So x with the bar, that equals the mean or the average. The mean is the same as the average. So this is our hypothesis. Our alternative hypothesis is that the means will not be the same. And we'll make a prediction that something like, if temperature is higher, then the animals will increase in speed. And then we have to qualify that with a because. So an experimental hypothesis is usually written in the if, then, because format. If temperature is higher, then the animals will increase in speed because they are looking for an ideal temperature. Or they're something like that anyway, something like that, if temperature is higher. So if, then, because. So that's about the means. Now about the choice experiment. Well, in essence, for a choice experiment, the hypothesis, the null hypothesis is the animals have no preference. Of whatever it is you've decided to test them on. And your alternative hypothesis will be the animals prefer whatever it is you think they would prefer because for whatever reason you think they would prefer it. So you state your hypotheses. So what's really important is that these hypotheses must be testable. You must be able to test them and you must be able to measure some kind of response. So you'll have to decide on what that is. So for your measured response, so question 1, measured, we've already looked a little bit at design, so how you'll design your experiment. Um, so wait, first of all, you have to decide. So you must decide. Will you do a choice experiment or response experiment? So that's one decision that you need to make. You decide on your a variable of interest. Is it temperature, salinity? We can measure a lot of different things. What else can we measure? We can even measure oxygen dissolving. We have the equipment to measure that. pH, light. So it could be something like light intensity or different wavelengths, as in the brine shrimp experiment. So we can do that in our lab as well. We have cellophane that can create different light wavelengths. Uh, so you have to decide that. And you have to decide the um, measurements, not the measurements, the not the quality, no, that's not the word either. What exactly will you measure? Or no, what will treatments be? What will treatment levels be? That's what I wanted to say. What will treatment levels be within the tolerance of the animal? So you don't want them to die. And decide on the number of animals. If, for example, you have a choice experiment, question 2, choice experiment, you should use 15 animals at the least. Altogether. So if you have 3 choices, you should have 5 animals for each choice. So if you had 4 choices, you would need 20 animals, but 3 choices is lots. So for example, you've got a large dish and you've divided it into 3. As you can see, exactly even. They should be exactly even. And you can test the animals on, for example, color of substrate. So maybe this substrate is red. Substrate is the bottom on which the animals exist. This one is blue. This one is black. So those are your choices: red, blue, and black. So you'll be placing your animals in the center one at a time, observing which choice they make, and then taking the animal out. So you have to also decide how long will you give them to make that choice. How long will you give the animal to make the choice? So another aspect of a choice experiment, so we're trying to be as random as possible. So you should choose your subjects randomly, as randomly as you can. So it could be that the animal chooses whichever choice it's faced with first. So if it's facing this way, it'll just choose that one. If it's facing that way, it might just choose that one. So you need to orient the animal in different directions and choose those directions randomly as well. So place the animal in center, facing different directions. I think a lot of people are using the crab, so that will be good. Yeah, so that is for a choice experiment. It is good actually to use animals of about the same size if you can. Um, okay, so you need to fill out, after you figured all this out, page 57. That is called the outline of your experiment. Page 57 in your lab manual, and it's called Outline of Your Experiment. I'll show you what it looks like. And so you can, I know, sorry, I didn't leave that up very long, but it is in the, not the lab manual, we don't call it the lab manual. It is in the activities guide, page 57. So once you fill out the outline of your experiment, then you can start preparing your proposal. You know, one of the reasons we do this in this class is because when you get into science and when you get into doing research, you're going to have to make a lot of proposals. Because for one thing, you want them to be accepted, say, by a professor or a research associate, but also you might have to make proposals for funding. So making proposals is a big thing in science. So your proposal to the class is on pages 63 and 64. But you're using the outline of your experiment to put together your proposal. Yeah, so you can do this in PowerPoint or a similar program. And in that proposal, you're going to talk about your animal. You'll show some pictures of it in its natural environment. You'll talk about some background information about its habitat, the variable that you decided on, your hypotheses, and your experimental design. And the reason for that is so that the other students can see what you're doing, but also so that Robin and I can see how you've designed your experiment and give you some feedback. Because we've been doing this for quite a long time and we can help you to get the best results out of your experiment. So the proposal. So you're doing the proposal. You can do it on your own if you wish to, or with a partner. So I'm trying to contact people individually to see which you would prefer. So I'll keep working on that. I just got your literature report, so I'm sorry I haven't been able to read them yet, but I did glance at them and what you're working on. So I might pair up some students. So the proposal can be on your own or with a partner. So if you have a partner with that, you put it together and present it together as one. As one. Of course, it will be a little bit different than in the classroom. Everything in this activity guide is based on we'll be in the classroom. We won't be in the classroom. We'll be on Zoom, essentially. So um, it's not that different, really, though. You can show your experiment, your proposal. We'll put it up on a share screen, or I'll put it up on a share screen, and then we'll set it up in such a way that you can talk about it while we go along and get some feedback. So, how to collect your data? Conducting experiment. Since we're in this pandemic situation and you won't be able to work closely together, you'll have to decide which individual does which part of the experiment. So you will be working on your own, but if partnered, can divide the data collection. So there are two good areas in your activity guide. Let me just find the pages for those ones. They're very good. So starting on page 59, there's an example for response experiment on page 59, which shows how to collect your data, how to put it into a table. And on page 62, for a choice experiment, on page 62. So take a look at those. I hope that you have your activity guide with you. What I'm going to do now is I'm going to sign off and I'm going to get you to take a look at those on your own. I could share it, but I don't think I need to share it. I think that you can take a look at those on your own in your activity guide. And then when you come back from that, it will be 4:30 and we will have Robin show us some of the equipment that's available for us to use in the lab. Okay, so I'm going to sign off now.